will be Neil. He's received his bachelor and master in physics from India, and his physics PhD from the University of Texas in Austin. And he works with computational and visual neuroscience. After that, he continued a postdoc also in the University of Texas in the Center for Psychedelic Research and Therapy. And now he moved to Brazil to start his second postdoc here at the Braulio's lab, analyzing clinical data as EEG, biomarkers, which he will present today, and bringing psychedelic therapy into Brazilian public health system. So thank you, Neil.
Is this working? Okay, so I found this out recently that Brazil globally ranks, let's keep that light turned off because it reduces the contrast of the slide. Thank you. Ranks 5th for the prevalence of depression cases and 1st for anxiety. And 30% of the depression patients are treatment resistant, meaning that they do not respond to at least 2 of the kind of top standard pharmaceutical antidepressants. But the other interesting thing about Brazil also is the existence of this psychedelic beverage called ayahuasca, which is a traditional Amazonian brew that is mixed, that is a mixture of 2 plants. And recent research, including in Braulio's lab, has shown that ayahuasca holds a lot of promise in treating this treatment-resistant depression. So about 10 years ago, in the Braulio lab, there was a clinical trial of ayahuasca for treatment-resistant depression. And there were 46 healthy people and 29 with treatment-resistant depression. And each of these 2 groups were further subdivided into an ayahuasca group and a placebo group. And around 24 biomarkers were collected from these people before and after the treatment. And the kinds of questions that we are asking using this biomarker data are the following. The first is, how do these biomarkers differ between the healthy population versus the people with treatment-resistant depression? And how do the biomarkers change after an ayahuasca versus a placebo dose in healthy people? And the same question again in depressed people. And finally, which biomarker changes correlate with improvements in the depressed people as measured by their subjective self-report scores? And for today's talk, I will be focusing only on the first question, which is how do the biomarkers differ between the healthy and the depressed people at baseline before any kind of treatment? Okay, so 3 papers have been published already about the role of individual biomarkers in this question. One on salivary cortisol, one on C-reactive protein, and one on BDNF. And these papers mostly look at analyzing individual biomarkers for this clinical trial. And what we are going to do differently here is not the analysis of individual markers, but to see what putting the markers together, analyzing their joint distribution can tell us about these questions. So some analysis has already been done by putting all of these markers together using a machine learning model called the random forest. And what you essentially do is that you feed in the information about all of the markers to the random forest, and it takes all the markers together and uses its discrimination algorithm to separate the healthy from the depressed population. And it kind of spits out at the end the relative importance of the different markers in being able to separate these 2 distributions. But the thing you have to remember is that it's not telling you individually how well the CRP does. It tells you in using the entire combination of all of the markers, what is the relative importance of each of them. So this is sort of the relative importance of the markers according to the random forest model. But what I'm going to tell you about today is a different way of analyzing this data that I've done that's not the random forest model, and at some points I'll draw some comparisons between the 2. So I'll first start with just a raw plot of what the distribution of these markers look like for the healthy and the depressed people. And you see here the blue is the healthy people, red is the depressed, and these are all of the 24 biomarkers. And the error bars show you the mean plus minus standard deviation, and the red is for the depressed people. And on top of that, we also have violin plots. And essentially what you will see is that these markers towards the beginning, the red and blue distributions are more different from each other. And as you keep going over here, the red and blue distributions become almost the same. So these markers towards the beginning are the ones that are most different between the people who are healthy versus the people who have treatment-resistant depression. And what I have used, so you can use these distributions to try to separate the two groups. And what I've done here is I've used a method called quadratic SVM. SVM stands for support vector machine, to use each of these markers individually. So far we're still talking about individual markers, to use each of these markers individually to separate the two classes. And these gray boundaries that you see are the kind of boundaries that it puts in the axis of the markers to separate the two groups. So for example, if you look at, let's say this one, this gray boundary means that the classifier says anything below this value of this marker you classify as healthy, anything above you classify as depressed. Some of the markers have single boundaries like this or over here, but some of them have double boundaries. And double boundaries, this is a feature of the quadratic part of the quadratic SVM. It can put double boundaries. As we will see, these double boundaries are very crucial. The double boundary here means that this classifier has put two boundaries such that if the value of this marker is in between these two, it classifies it as healthy, otherwise it is depressed. Okay, so using this kind of a classification scheme, we can classify the two groups, and at the bottom is the plot of the classification accuracy. What percentage of our data was classified correctly using this quadratic SVM on these individual markers. And these markers here, I have sorted them in the order of how well they can individually separate the two classes. So using, for example, just salivary cortisol, you reach a classification accuracy of about 76%. And as you keep going down, they become crappier and crappier in separating the distributions until you're almost at chance. Okay, so now let's talk a little bit about just the top marker, which is the salivary cortisol. Here is the plot of the individual data points. Blue is healthy, red is depressed. And what you see, and has already been commented on in the paper on salivary cortisol, is that multiple studies have shown that depressed people have lower average salivary cortisol, but they also have a higher spread. So that means when you're looking at the depressed population, some of them have both lower salivary cortisol than the healthy population, but some also have higher. And the sort of intuition, I guess, about this that you can say is that cortisol is known as a stress hormone, which makes it sound like a bad thing. But when you wake up in the morning, your body releases cortisol, and this provides drive, you know. If you have too little drive, which is seen sometimes in depression, you have like fatigue. You can't go about your day. But if you have too much, then you have something like a neurotic anxiety or stress. So depression is kind of like a bucket term. Sometimes you see too low cortisol. Sometimes you see that it's too high. And if we now use our quadratic SVM on this cortisol values, what it essentially says, is a bit of math, but anyway, basically it says that you should classify a person as healthy if this quadratic equation is greater than 0, where x is the value of the salivary cortisol. The Q2, Q1, and Q0 are numbers. You don't really need to worry about what these numbers are, but essentially this is a quadratic equation. And if you remember from high school, a quadratic equation has two roots. Essentially what this is saying, these are the roots 333 and 1185, which I've drawn over here. Essentially it's saying is that if the salivary cortisol is in between these two values, classify as healthy, and if it is outside, classify as depressed. Okay. So, I mean, here I would like to draw a comparison to the random forest algorithm. The random forest algorithm is essentially a whole bunch of decision trees. When you give it a data point, it puts the data point through each of these decision trees, and each of them outputs a decision, healthy, healthy, depressed, 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 healthy, and you take the majority of the votes and say, okay, it's depressed. Because of this reason, the random forest algorithm is a little bit of an opaque black box. You know, its decisions are not very easily human interpretable. And if you have to explain to someone why your model gave you this result, you can't really sit down and explain to them. You basically have to give your black box model to them and they just run data through it. In comparison, I believe that this kind of a model is much more transparent. It's not a black box. And the classification rules it gives are more interpretable, and it allows you to have a discussion about what these markers are doing, and it yields more insight. Okay, so let's see what's going on here. All right, so now that we have talked about the top marker, let's add the second best marker. The way that we do this is we were doing the classification in a one-dimensional space, and now we are doing the classification in the space of 2 dimensions, where each of the dimensions is a marker. So we have the top marker here, and what we do now is we pick each of the rest of the markers and put it in the second dimension, and use this quadratic boundary again to classify healthy from depressed and see which second marker allows us to have the best classification accuracy when we are in these 2 dimensions. And what we see is that LDL cholesterol is the one that gives us the best classification accuracy. The boundary for classification here is still a quadratic, which describes an ellipse. And here is the equation of the quadratic. Q2, Q1, and Q0 are no longer just numbers. They are matrices and vectors, because this is happening in a 2-dimensional space. And X over here is a vector of the numbers of salivary cortisol and LDL. So it's a vector with 2 numbers, the top 2 markers. And now the accuracy jumps from 76% to 85% if you use both of these 2 markers together. Okay, so what is this LDL cholesterol? I found out a little bit that when the body is under stress, it releases energy into the blood. But if you're not actually using that energy for something, then it just kind of hangs out in the blood. And it accumulates as LDL cholesterol, also called kind of like bad cholesterol. And I think I'm just going to use this. What we can also see is that if you looked at the individual accuracies of all of the markers a couple of slides ago, CRP was the second, C-reactive protein was the second best individual marker. But here, when you want to combine it with the top marker, LDL cholesterol narrowly beats it. So CRP is no longer chosen as the second best marker in this multivariate analysis. And the reason for this is that when you introduce a new marker into this picture, what counts is how much information this new marker can bring in that has not already been accounted for by the existing marker. So by itself, a new marker individually may have good classification accuracy, but if that information was already contained in the markers that you already have, then this new marker is not bringing in too much. So for this reason, LDL beats CRP, because it brings in new extra information that helps to discriminate the 2 classes even better. And this is an example of how we see that combining features together in the same space sometimes gives different and surprising results than just looking at the individual performances of the markers. And once again, if you compare to the random forest model, this is a very transparent model where you can look at the picture of what is happening in the joint space of these markers and how they're interacting in the decision space. This kind of a picture is not very easy for the random forest model to give you. Okay, and then we add a third best marker, which turns out to be the ratio of 2 markers called AST and ALT. And once again, this is happening in the 3-dimensional space, but the classification boundary is still a quadratic. It is trying to separate the healthy and the depressed points. And you can see most of the blue points are inside and the red points are outside. And I read up a little bit about this marker, and I see that it has been shown that depressed people have a lower value of this ratio, and it is associated with a stressed liver. And the fact that these markers that are showing up in discriminating healthy and depressed people are not just neurochemical markers having to do with the brain, but they have to do with what's going on in the rest of the body. The AST-LT ratio has to do with the liver, for example, is an indication that what we are looking at for at least some of the people is signifying something called immunometabolic depression, which is not just an affliction of the mind, but kind of like an overall stressed body that's affecting different parts of the body. Okay, so now that you have an idea of how we are adding marker by marker to increase the classification accuracy of the healthy and depressed people, we can keep doing this one by one for all of the markers. And the kind of purple line here is how we keep increasing classification accuracy. And so this is the ordering of, if you do this, what I'm calling greedy accumulation, because in each step you pick the marker that most increases the classification accuracy. So if you do this greedy accumulation, this is the ordering of the markers. And the black curve here is the plot of their individual accuracies that I had shown before, just in a new order this time. And the gray curve is the incremental accuracy, meaning when you add LDL, for example, how much extra accuracy are you gaining by adding this compared to the collection of markers you already had before? And so what we can see is that after only around 6 markers, you get 100% accuracy in separating the healthy and the depressed populations. The other thing that you notice is about CRP. The random forest model had said that the CRP was the best marker, even when you put all of the markers together. In the initial analysis, it had been seen to be the second best individual marker, and a whole paper was written about CRP. But in this joint analysis here, we see that it's not even in the top 6. And once the top 6 markers have taken care of 100% accuracy, the rest of the markers are not even that useful. But one caveat I should mention is training performance versus testing performance. These classification accuracies are being measured on the training data, meaning on the data which was used to train the model. So a model always performs better on the data that was used to train it, and you don't expect the model to have this same performance when you now give it new data, like when it performs in the wild. So these classification accuracies are sort of optimistic. You expect the model to perform worse when you actually give it new data. And for that, I still have to do this. It's called cross-validation, where you train the model using 80% of the data, and then you test it using 20% of data that it hasn't seen yet, and see what the testing accuracy is. And that gives a more accurate idea of how this model would be expected to perform in the wild. So just keep that caveat in mind. Okay, something else that we did. So these methods that I'm talking about are ones that I developed during my PhD and postdoc in the US. And one of the things that we did was a method where, imagine that you're in a multidimensional space, like a space of 3 variables where you're discriminating 2 classes. We came up with a method to condense those 3 variables down to 1 variable to classify the 2 classes without losing any classification accuracy. And that's what we are showing over here, is this sort of unified marker where you go from a multi-dimensional space to a single-dimensional space. It's basically a form of what you would call dimensionality reduction, like PCA or something like this. So in the beginning, when we only had salivary cortisol, this unified marker is a function only of salivary cortisol. And then when you bring in LDL, this unified marker is a function of these top 2 markers, but boiled down to a single marker. And then this one is a function, this one is a unified marker, which is a function of the top 3 markers. So in each case, you can boil it down to a single marker and classify using just this single marker, and the classification boundary is just 0. Anything less than, above 0, you say healthy. Anything below 0, you say depressed. And you see, as you combine more and more markers into the single marker, the classification accuracy of this single unified marker keeps getting better and better until this single marker, once it has merged the top 6 markers, by itself can classify with 100% accuracy. And so if you use this method, you can kind of call this a single marker for classifying healthy and depressed, but of course this is a function of several biomarkers, but it is a known function. Okay, so in summary, I would say that we analyzed biomarkers collected during an ayahuasca clinical trial. Using support vector machine, we showed how well they distinguish healthy from depressed, both individually and when combined together. And this is a transparent method that yields insight. And among future work, I would like to apply these same methods to the other questions, because I only talked about answering one of those questions. And the other questions have to do with changes in these biomarkers after treatment with ayahuasca or placebo. I would like to thank Fernanda and Ignacio for helpful discussions regarding the processing of the data, and Sergio for helping me prepare for the talk and giving me feedback, and also the USONA Institute of the United States for funding my research here. And with that, I'll take questions.
Questions?
So Neil, thanks for the great talk. I mean, it was calling my attention, I was mentioning with Rodrigo this necessity for having something for training and something for testing, but you already mentioned that, so I think you are well aware of this, the necessity of this cross-validation. So then I'm gonna make another question. I mean, this would be the original one, but you answered. My question regards the ordering, you know, because as far as I could tell, so you have cortisol and then LDL, and anyway, you have the accuracy increasing as you combine more and more of these boundaries. But is this dependent, so in here CRP was 6, but what about if you change the order in which you start grouping them? Can't you see a different picture?
Yeah, that is correct. I think you guys can hear me without the mic, right? Yeah? That is correct. Yeah, really the best way, the most conclusive way is to take all possible combinations of 3 markers from the 24. So that's 24 choose 3, and this is an NP hard problem. You have to really do it like brute force, evaluate all of the markers. And it is possible that the greedy algorithm, which takes the best marker first and then the second best, does not necessarily give you the best combination of top 3, but it is kind of a sort of lazy way to try to get to some answer that is probably close to the optimal. But the best way is to take all possible 3 markers, which it gives you a kind of like a combinatorial explosion. If you have like 24 markers and then you have to run this on all possible 3, then it just takes a lot longer. But it is possible to find, potentially it's possible to find some other 3 markers that give you actually better classification than this top 3. So yes, that is something that is true. And this is a kind of problem that affects other classification algorithms like random forest as well. This is the reason why the random forest is not a single decision tree, but a whole bunch of decision trees, and then you kind of take an average of those, because it kind of creates all of this sort of like random search paths. And in order to come to a robust result, the thing that I would have to do is it's actually tied to the cross-validation. So I would take 80% of the data each time, but a different 80%. And then every time you take a different 80%, this greedy algorithm would come up with a different combination of the top 3 markers. And then later on, you do an ensemble of these and see how many of those markers were chosen how many times in the top 3. And that gives you a little bit more of a robust answer than just using one greedy algorithm.
Hi, hi, thanks for the talk, great insights about it. I was wondering, you already mentioned in your talk that there are a lot of different kinds of depressive patients, right? And the top two markers that you show are related to metabolic syndrome. So I was wondering whether first, have you considered the body weight or a relationship between body weight and height, whether it will fit, because body weight is very easy, just you need a scale and then you can say something, and whether you can separate depressed people with and without metabolic syndrome using this technique?
I didn't totally understand your second question.
Well, let's see, because I believe two of your biomarkers, or the two measurements that you show, are associated with energy consumption, right? So the ALT and AST are associated with liver, as you mentioned. LDL is also associated. So I believe these patients probably, I would presume that they are high, they have high body weight. So meaning that probably they're, well, it's difficult to establish a causality, whether they are depressed and therefore they exercise less, they eat more, or the other way around. But I was just wondering whether you can, using this, using the body weight, you could separate between two kinds of depressed patients.
So the first thing I would say is that I am not the best person to answer these questions, because at the stage that I started doing this analysis, these biomarkers were just numbers to me. I wasn't around when these biomarkers were collected. So the people who collected this, for example, Fernanda and others, have a lot more intuition about why they decided on these biomarkers and what they saw in the patients in real life as they were collecting the markers. I don't know if they collected body mass index or height and weight and things like this. She says that she did. But my intuition about these markers is, I would say, kind of limited now. It was only after I started analyzing them, treating them as just numbers, and these markers started sorting themselves, that's when I became curious to see what are these markers about, what do they do? And what I would say is that in my little ChatGPT research so far, AST/ALT has shown up to not just be related to body weight, but also to what is happening in the body. Like there's like stress-related changes that are happening in the body. And also a lot of these people have been taking medication. And when you take antidepressant medication, the liver is affected, and that also kind of changes the AST/ALT ratio. So that is kind of interesting, because it's not the medication that has caused the depression, but you will still see a correlation between people who are depressed and people whose bodies have certain kinds of markers because of medication that they have taken. So I think the picture is not very clear, and I guess you could try to separate between the two groups. Oh, by the way, the other thing that I found out is that people who have early depression, in the beginning stages of depression, their salivary cortisol is higher, so you're kind of more in an anxious state. But as you keep having depression for long periods of time, years, then it's like the nervous system becomes kind of like numbed or blunted, and then you have lower cortisol than average. So you get both ends, and they're kind of classified as two different types of depression. So the entire picture, in my opinion, is complex, and the story is not very simple about what is going on. And I'm not entirely sure with our sort of limited data that we can fully go into like stratifying the cases, or for this, at least in the case of this analysis, just kind of look at the whole statistics. Yeah, I don't know if that's a satisfactory answer to your question, but that's kind of all I know.
I was just curious about one methodological part, that is, if you choose this machine learning technique during the force for specific reason, for the nature of the problem, or for the nature of the data set, like, if you could have used it, I don't know, neural networks or another kind of machine learning techniques, and why did you choose the random forest specifically in the VSM? And I also was curious about if there was another method for evaluating the model, because or if it was only based on the accuracy, because like I know that maybe could be some difference, for example, if the data set was balanced or not, and then it would influence in only seeing the accuracy. So that's basically my question.
Yeah, those are all good questions. So the random forest analysis was done by a group of researchers before me. I'm not the person who did the random forest analysis. And when I asked them why they picked the random forest, one of the reasons that they picked it is that among the different classification algorithms, random forest gives you this sort of like a priority list of the rank of the variables and how much is each variable important. So that's the answer I got from them. I personally used this quadratic SVM because I have had a lot of training in this kind of Bayesian classification methods and things like this. So for me, these methods are a lot more intuitive, and I can understand not only what the model is telling me, but why it is telling me. So I felt like the quadratic SVM is something that is pretty well suited to this problem. And the other thing that I'll mention is that there was a class imbalance. As you mentioned, there were more healthy people than depressed. What I have found out is that the random forest model was not designed to, in this analysis, was not designed to take that class imbalance in account. So it would have to be corrected to take that class imbalance in account. But all of the results that I showed you using the quadratic SVM, the accuracies that you saw, for example, were class balanced accuracy, meaning it wasn't looking at the raw number of red and blue data points that were classified accurately. It was looking separately at what percentage of the blue data points was classified accurately, what percentage of the red data points were classified accurately, and average the two of them, meaning you're trying to estimate the classification accuracy if the data came from two populations of equal numbers. So, so that heuristic has been taken care of. Here.
Thank you.
Yeah.
Thank you, everybody. So, thank you, Neil. Now I want to invite Rafael Lima. Rafael has a bachelor degree on biological science and master's and doctorate in psychobiology, with emphasis in neurophysiology, all from the Federal University of Rio Grande do Norte. He currently works as a postdoctoral fellow at Brain Institute, and his studies to understand the cellular and molecular effects of DMT and its analogs on neuroplasticity. Thank you, Rafael.
Hello, thank you all for receiving me, and it's a pleasure to be talking about my research in the last 10 years, where I did my master and my PhD here in the Institute on the supervision of the Professor Richardson, and at some point when Braulio and both my mentors start to working together, and they are just amazing. I named this talk investigating psychoactive tryptamines as stimulators of neuronal plasticity with a focus on hippocampal neurogenesis, which is the process that I choose to study in this pursuit in the academy. And I just gonna do a very short introduction about what psychedelics is. This is a class of drugs that has been discovered and discovered by science, and it's ancient, used by many cultures. And they basically, the name psychedelics was coined by Humphrey Osmond, together with the scholar Aldous Huxley, and it's basically a neologism for the Greek that means mind manifest, because everything in the realm of the mind can happen when you are under of this substance. And my work is focused on psychoactive tryptamines, and especially the NN-DMT and 5-MeO-DMT that I did my master and then my PhD. And then they are called classical psychedelics because they work through serotonergic ways, and mainly through that. And then we have another class of drugs that can induce similar effects, but they are called atypical psychedelics because they do not work through serotonergics. And just little recap, theoretical recap, so we can understand. It's that the whole idea of my projects circle around the motivation of understand better and treat better depression and related mental illness. And then we start with the legacy theory about the neurogenic hypothesis of depression, which is meaning, it's basically, it says that waning and waxing of neurogenesis in the hippocampal formation are important causal factors respectively in the precipitation of and recovery from episodes of clinical depression, meaning that you can understand when the person is going in a depressive episode or going out of depressive episode, accompanying the neurogenesis in the hippocampus, the adult neurogenesis, sorry. And the mechanism is very simple. It's basically down serotonin, down neurotrophic factors, in this case the brain-derived neurotrophic factor, and then you go down to proliferation. And this gonna mean, brings a lot of comportamental symptoms. But it fails to explain the rapid efficacy of ketamine that was like the big breakthrough in the treatment in the last 10 or 20 years, and other psychedelics that act as like it. And then the hypothesis has been updated, and it's incorporating a lot of things that has a lot to do with things like Neil said once before, there is some core cytokines and other inflammatory markers that may be working and involving in the understanding of what is happening in depression, because it's basically updated the theory with the idea that chronic neuroinflammation can be somehow causing or supporting the symptoms. And it's basically when you have a prolonged cytosine activity perturbs multiple neuronal functions, not limited to neuroplasticity through microglial activation, but also neurotransmitter signals help take and release. So it's basically homeostasis. You are perturbating the homeostasis. And this happens when you have a lot of cytokines, then the blood-brain barrier got messed up, and other things start to go into, and it's also some peripheral immune cells and other signals that should be out of the brain, they go inside, and this can make things worse. And the key improvement of this idea, the general idea of put the inflammation inside of this understanding, is that it changed the focus from solely neurogenesis to expanding it to neuroplasticity and other homeostatic conditions for transmission, neurotransmission. And just to finish the introduction, I'm gonna talk about the cells that are produced in the adult hippocampus. They are the granular cells. They are, it's like the only niche in the human brain. There is other animals that has another niche, but where they are produced new neurons along the life after the birth. And then these neurons, like they probably encode, you need to believe me because I do not have enough time to explain that in details, but if you find me outside, we can talk. But it's just basically the granular cells and THRs participate in the process called pattern separation. When you have similar engrams and you have similar context, they can be separated by different memories and people hypothesize how is it, but some people say there is a timestamp or other kind of things that the granular neurons add, but they majorly participate in this process when you have similar contexts and you separate them. The opposite of this process, it's make similar memories or similar engrams, the same thing. This is going to be important. And in the first experiment that I done in my master, the basically idea is, if we put the 5-MeO-DMT, a psychedelic that it is anecdotal linked to treat depression. Like it's not in the academy, it's not working like as a medicine yet in that time, like back time from long time ago. And then we just understand that maybe could be about the serotonin signaling, and the serotonin signaling especially works well with plasticity in the hippocampus. And then we find, okay, it can work. And let's see if we can alter this process in health subjects. And then we go for other questions. And then we just inject some mice with 5 mg/MT, and then look for difference in cell proliferation and other morphological changes, but also functional changes. And this is the first experiment that I did here in the institute, not actually here, in the other building. But we basically showed that the animals that were treated with 5 mg/MT has a lot of new cells marked by the bromodioxyuridine marker that is just marking DNA synthesis. So it can be a lot of stuff, not exactly neurons. And then we did another experiment with another protein that is the DCX, the double cortin. And then we showed that the 5 mg/MT animals also got a lot of more cells, and it's like almost a month after, and this is like right after the injection, and this is about a month after, and you see that it still has more cells survived, like for that time. And then we start to analyze those cells, like for the process, and we see that the neurons treated with 5 mg/MT has more branches, which can be a signal of more integration in the circuits, because, well, neurons that wire together fire together. And then we have show that the 5 mg/MT has more branches, so neurons are more complex, and we control these populations to know the exact age of these cells. That was the whole idea of using the genetic model that we have. And then the cells was around like they were teenager cells, and we were compared them, and they were equal in time, but not equal in morphology or function. And then we go to patch clamp experiments where we compare those groups, the saline treated and the 5 mg/MT. Remember that these animals are healthy. And then we show that at the same speculated age of the neurons, the 5 mg/MT treated neurons are more close to what adult granular cells are. Yeah. And we compare a lot of electrophysiological properties like passive and active, and do some protocols with them, but then we just bring the ones that are different. That's about it. And then we found that the AP threshold for the cells was lower, more close to positive for the 5 mg/MT. And also the first after hyperpolarization duration was slower also for the 5 mg/MT, which also had a bigger instantaneous frequency, meaning that the cells could fire more in less time, and also a lot of spontaneous activity, which is a marker of the cell receive projections of the older cells, so it is more connected for the 5 mg/MT. And then we finished the first experiment. 5? Cool. And then I set off to a new start, and then I think that I have other questions to ask about, and this is going to be answered by a depression model, animal depression model, since I'm working with mice. And it's just the UCMS model of depression. It's one that I choose because it mimics better what happened with people in the real life. There's just a lot of stress and shit and everything with the animals, and the animals just, well, suffer like for a long time, and then they develop depression. So, well, looks like life, right? And then I choose these animals, and we apply the model on them. Like, the experiment is very long. If you are interested, we can talk later, but I just want to show the results here. And then the animals go like for 2 months in the treatment to develop depression, and we show here by the score of the cold state of the animals, which is a marker of selfless care, take care of himself, like the grooming. And then we also take note of the weight in the animals, and then we show that the model did work. Animals from the UCMS group the ones that received the treatment, like, did not gain weight, and also did not, also had anedonia state installed, like we did from the sucrose preference test. And then we show that the animals just have got the depression-like symptoms. Okay. And then we start to test these animals. And there is like a lot of groups, but it's basically animals that receive the MT and also animals that receive the positive control, which is the fluoxetine treatment. And then we introduce other groups, like the other questions that we had, like one group that received DMT under anesthesia, and other group that received DMT in the middle of the process of develop depression. Just questions that we had. It seems complicated, but it's very simple, at least in my mind. The first test that I did with those animals was the delayed non-matching to place using the radial maze. It's the test is basically to sample the pattern separation. That is the function that I say to you before, like the granular cells are doing. And it's basically, you put the animal in a very similar context, and then you just keep increasing the distance between those arms, and then we'll need to differentiate the place that he already visited, and there is no reward, and the new place. And you just increase the distance of the arms, and then as long as it is further, the animal find it easier. And it's closer, the animal find it hard. And it's a sample of the hippocampal neurogenesis. It's very well established. There is a variation of this test that you can do even with humans. And we just showed that the animals that receive the treatments, there is the DMT with anesthesia, or DMT in the middle of the fluoxetine, or the fluoxetine, which is the positive control. The animals did perform better in the task where it matters, because the S2 is the harder one, when the arms are very close to them. So in this context, the animals that has normal pattern separation, normal adult neurogenesis in the hippocampus perform better. This one doesn't matter because it's not hard. They always find. And just I put some data organized to see the rate of learning, but we couldn't actually take in any conclusion of this data. But as you see, the fluoxetine treated animals has a steep slope of learning, while the other ones, I don't know, something has happened. But just that. And then also I did other experiments, behavior experiments. I did 6 behavior experiments, different behavior experiments. And I did an open field and, I did an open field and elevated plus maze just to see anxiety and other exploratory behavior. But we did find any difference among the groups. And we speculated it a little bit, but I think that I need to go faster. And then in the same article, I just counted the whole number of cells, like I did in the master, but with the different conditions, and we found some interesting differences. And also one new thing that I bring in the analysis is just that the fluoxetine treated animals had a lot of ectopic cells, which are cells that are with normal morphology, or at least identifiable normal morphology, but they are always very outside of the place where they should be. And we do speculate a little bit about that in the article. And this is also other thing that I produced in these years. There is a small review about the psychedelics on neurogenesis and broader neuroplasticity. I did expand it a bit, to get the grasp of the field. And that's it. And this is the people that I need to thank. There is the lab that I've been developed, and this is the new generation of the lab that I come back after some time, and I'm loving it.
Thank you, Rafael. Questions?
Hi. First, I would like to congratulate you for your recent paper. translation psychiatry, and also for your outfit, super nice. And but the thing I was asking, you know, like if to fully stop neurogenesis after the injection, right, would be hard for you to separate what is really related to neurogenesis and the improvement of behavior. And I was just thinking an experiment, if we could maybe after your injection, that now that you know more or less the window where there is this boost in neurogenesis, if you could, you know, like in that time, induce, for example, I don't know, like a dread, or dread would be better because then the hippocampus is huge. And then like to check with this behavior, like continually, you know, inhibit those only cells that are new that use dread, if you think that that could work, you know?
Well, that's a very nice idea. It's like we should like do a different insult with using some manipulation technique that we think that. Mm-hmm. And then we compare like with artificial way of manipulation, like optogenetics, dread, and other things. It's a very great idea. I think that, but well, now I learned that when we put a group, you need to like to put the proper control, and then the proper control can take another control, and this can make things messy.
I have more eagle's eye question. So I'm not an expert here in neurogenesis, but when I talked with people like 10 years ago, I got the impression that in rodents it's much more pronounced and rarely in humans. So what is the current assessment of how much the human brain is doing neurogenesis in hippocampus? And perhaps that also relates to the question, so what is the effect on the old neurons and on new neurons? Both might be interesting.
Well, this is a very pertinent question. It's actually like a big discussion in the field. That is the relevance of the adult hippocampal neurogenesis to humans. And the data that we had like until 2018 and something like that was that in humans it goes down when you age, and it starts to be less relevant for the whole physiology of the brain. But this whole idea was centered in data that was collected from patients like in ambulatorios, I don't know how to say that in English, in hospitals. And these patients was like very, like a lot of other conditions and other things that comorbidities, and well, when they die, like they need to process the tissue and go forever a lot, and takes a lot of time. Then you need to put other things to reveal the DNA synthesis or other things like that are related, like signaling or new generation of cells. And I think that in 2022 or 23, I can't remember the author, but they like didn't show like we couldn't find this level of neurogenesis that we find in rodents, in humans, but he did show that the methods that was used so far and the process that take times from the tissue of the human brains degrades the way to find, you see? So it's not evidence that it's relevant for humans, but still we need to find other ways, because we can just open the minds and the brains and remove the tissue like we do with the animals when they are young, and not when they are aging, dying, and that's what we do.
Thank you, Rafael. I know that some people has questions yet, but I will ask you to do the questions during the coffee break, okay? Yeah. Thank you. So our next speaker is Aline Elvina. Aline graduated in pharmacy from the Federal University of Rio Grande do Norte, and she's about to start her master's. She is about to start her master's in the neurodynamics laboratory. Thank you, Aline.
Hello, everyone. I'm Aline. Wait. So my project is about a smart camera for automatic respiration, reflex estimation in neonates. It's my supervisor is Richardson Leão, with collaboration with Elton Maia, Simone Ribeiro, and Silvana Pereira. So a lot of signals and vital signs we have, it's very important to newborns, and monitoring the signals like movement pattern, respiratory rate, and in the ICU, neonate intensive unit care, these signals are monitoring by sensor. They have to be in contact with the skin of the baby, and they mainly are belt in the baby, and these sensors cause skin irritation. We can see in the picture with this article, but and these signals they need to be in contact with cause irritation in the skin of the baby. And this article, the skin interfacing wearable biosensors for smart health monitoring of infants and neonates, they bring a skin sensor, but they cause less irritation than the usual. But what you want to do is try to do this without any contact with the skin, with a smart camera. So we doing this with a camera, so we hope the babies can find their respiratory rate and movements without contact the babies. So also primitive reflex are one of the things we're trying to monitoring, like motor reflex, walking reflex, rooting reflex, tonic neck reflex, grasp reflex. All these reflex are very important, and some of these reflex come and go until the first year of birth, the baby. So monitoring the reflex is very important to do how the motor cortex development and detect changes in the brain of the babies. And we are correlating this primitive reflex and respiratory rate with another device like EEG. And the EEG movement artifact is a problem that we have when you are using an EEG. So when you use EEG with a camera, we can find and see what, identify what is EEG movement artifact, or what's the just a movement the baby was making. So we are using you look once, YOLO, and Eulerian video magnification are techniques by vision computation, and we are using these points we can see in the pictures around the baby's bodies, and trying to identify these movements and the rate respiratory. We have a database we have with Professor Simone, and we mounted this data set and tried to find the best videos, because we have a lot of videos, but some of inside the incubator and others outside the incubator, and different angles. So we try to find the best angles and the best image to try to do this project. And this is some image we have make segmentation with YOLO. We can see here in the line, purple line. We made a segmentation around the chest of the baby. I think 5,000 of frames, the images we use until now, but we have a lot of things to do. This is just preliminary results we have. And this is a video we have generated during offline experiments. We can see when the baby start the movements in the line, green line, and then the baby stay in not movement. And also the sinus respiration. They have a little delay, but we are trying to better our system. And some preliminary results, we have this individual score with frame difference, optical flow edge difference, and also the score movement combined, and heart rate respiration and the movement. In blue, we have when the baby is in movement, and actually wasn't in movement, and in orange we have when the baby is in movement. We can see the peaks are matching with when the baby are in movement. So this is just a part of the project that I want to do, and we have correlated this data with video EEG, camera, thermal camera, more noise sensor, and correlate all these data and try to find neurological disorders and sinus, and find the sinus vital without any contact as possible. But some of the device needs contact like a EEG, but we want to try to do this with less as contact as possible. So this is just the beginning of the work. I have a lot of things to do, but that's all I have. Thank you all for your attention, and thank you all the guys from Leon Lab.
Thank you, Aline. Questions?
Thank you for the talk. I have a question. Is there a theoretical limitation of how fast you can segment these camera images and concatenate the movie? Because if you want to use this as in the future as something which replaces the normal life support system, it has to be extremely fast, right? It has to be real time.
Yes,
so,
we have to do this in real time. Just what we have is just made in a database now. But in the future, we have to do this in real time. So have to be very fast, but I don't know how fast you can do this, because we now just have training the system.
Well, I'm just going to comment what Kirsty said, because there are no baby database for this kind of things. There is nothing available. Then now we got the ethics committee approved, so we're actually going to have our own database, because most of the images that we got are like very bad quality, taken with a mobile phone, and we believe that with a camera that is well placed, that will be possible to do. And the idea that to create this model using this YOLO is that it can be como é que fala? Embarcado? Embedded, yeah. It can be embedded in like, the smart camera hardware. So, in fact, we're going to be able to, the idea that if we have this model that are simple enough, you can apply to, like 10, 20, 100, esqueci também. Não, da coisa incubadora. Incubators. Sorry. And yeah, so the idea is like to create this model is to be able to embed it and put like, in every incubator. So like, and the cameras you use, like the problem is not really the frame rate, but the time of detection, right? And if you're going to use this kind of things alone, you have to be sure that what you're getting is respiration. So the first thing the second thing we're doing is to compare this with accelerometers. So that will be a part of it. And I like it. It's very well. I'd like to congratulate you for your talk and for your selection, right? That you were approved in our selection. And welcome to, sorry. Yeah, Okay, yeah, but in your probable approval.
Thank you, Aline. So now I want to invite Flora. Flora is a teacher with a degree in social science from UFRN, and she's currently a master's student here at the Brain Institute, where she researches how to adapt evidence-based activities for literacy to the classroom environment and the effects of school naps on learning how to read. Thank you, Flora.
Good afternoon, everybody. So today I'll be presenting some of my master's research, which is called Multisensory Activities and School Naps to Overcome Letter Mirroring in Early Literacy, Adaptation of a Protocol for the Classroom. So our goal was to take a protocol that was developed by Arzin Torres, a former student of our lab, that was able to double reading fluency in first grade children. That means when we're talking about reading fluency, we're talking about the speed of reading. And these activities were done in a school setting, but individually with the children. Our goal was to have teachers, was to adapt these activities in order for teachers to be able to apply them in their own classroom. And why are we talking about letter mirroring? Letter mirroring is a natural phenomenon that occurs during the acquisition of abilities of reading and writing. What you see here is a student, a child around 6, 7 years old, which is the age range of the children that we work with. And what you can see here on the letter E is that it's mirrored, right? So it happens both on writing as well as on reading. And we believe this happens because some of the areas here around the fusiform gyrus, they are part of the visual cortex that recognizes specific kinds of images, right? So you have the FFA, for example, the fusiform face area, so it specializes in recognizing faces. And our brain, well, we are not born with the abilities to read, and what we found is that the region that specializes in recognizing words as such, so the visual stimuli for words as words, and of course letters, seems to be a specialization of these areas that they present this phenomenon of mirror invariance, which basically means that these parts of the brain, they recognize images, mirrored images, as the same representation of this thing on the brain. And because the VWFA seems to be this specialization of these areas, it would seem that it inherits this phenomenon of mirror invariance, which translates as letter mirroring. And this is a problem, of course, because especially with the lowercase letters, when you mirror some of the letters, the words become different entirely. This is not, of course, the case for all of the letters, but it's still an obstacle for reading. And our hypothesis is that the sooner we break mirror invariance for letters, the sooner we unlock other reading and writing abilities, such as fluency. And why to bring school naps in this experiment? So what you see here is a figure from the paper where the original protocol was published. And these are the error rates for the visual task, which I will explain in details later on. But basically it measures how well a child can discriminate between mirrored letters. And what you see here for the 3 replicas, so 3 different classrooms, and here you have the error rate for this task, is that both groups who went through the activities right after the intervention, they had a significant and great decrease in the error rates. However, 120 days after, only the groups that had the activities and slept after lunch, they kept the same rate, and also actually got a little better in one of the cases. But the group that went through the activity but did not sleep had an increase of this rate. So this is to show the potential of bringing the napping to the school schedule, and which was what we aim to do with this experiment. So for our research, we worked with 6 different schools in 4 different cities, and 3 were assigned to a control group and the 3 others to the intervention group. Our control group was a business as usual control. That means that apart from the moments where we were testing and this activity here, which I will explain, these 3 schools follow their regular program, the activities the teachers would already do with their classrooms. Whereas the intervention group, during 3 weeks, they had 20 to 30 minutes of multisensory activities in the morning, and then after lunch, they had up to 2 hours for napping. So to establish baseline, we went the week before to test all of our measures. We did that right after the intervention as well, and we did it, repeated the measures 90 days later, which is the end of the school year, to see what happened with these memories later on. And of course, for the intervention groups, we had a teacher training, so the teachers were able to apply these activities in the classroom. And because our activities, are done with lowercase letters, and lowercase letters are not usually taught in first grade for a number of reasons, we had a 20 to 30 minute lesson about the lowercase letters with both groups. And that was in order for them to be introduced to the lowercase letter if they didn't know it. And we did it with both groups, of course, because any results we would see different between the 2 groups. If one of them hadn't done the alphabetical lesson, we could attribute these results to one of the groups not having gone through this lesson. And these were our activities. So we had 3 of the 4 original activities of the protocol. The first one was writing on air, where children were blindfolded and the teacher would say the sound of the letter, and then the child would use their index finger to write the letter on the air. Then there was the writing on paper, which is the same reasoning, but instead of writing in the air, the children write with pencil on paper. And for last, the perception for action activity, where the teacher would show a letter like this, and the children would have to raise their hand depending on the orientation the teachers gave was you have to raise the hand the same side as the little belly of the letter. So depending on which side the teacher showed the letter, the children would raise their hands. And the reason why we had 2 of the activities done with eyes closed is because the mirror invariance seems to be a phenomenon of the visual system, of course. So the idea here was that children would create representations of letters without the interference of the visual system, and that would help break mirror invariance for letters. The last activity, it was done using the visual system, of course, the children were seeing, but we chose to keep this activity because it is designed for children that don't know the letters yet, so the children don't need to know whether this is a B or a D or what letter is it. They just need to know which side a specific part of the letter is, so it allows for children who don't know the letters yet to participate in the activities. And then, of course, after lunch, children had up to 2 hours for sleeping, and we measured the time slept and the days, the amount of days each kid slept by having a time schedule, a log, where the teacher or the person responsible for this moment would write down the time where the children would apparently fall asleep based on their movement, and the same thing for the waking up time, so seeing if they open their eyes or start moving again. These were the measures that we recorded for the whole research. We had mirror discrimination in a visual task, which is the one I'll be talking about. On a written level as well, we had a reading task to measure fluency, so words read correctly and the reading speed. And of course, the days napped. Today I'm going to be talking about these two measures. And to measure the discrimination between mirrored letters, we use a same different task, where each child would see a sequence of images that could be letters or symbols. And after seeing two stimuli, the child would have to press a key to say whether they saw different images or if they were the same. The stimuli could be the same letter or symbol repeated twice. It could be a mirror, a letter or a symbol, and then the mirrored version of it, or a completely different picture of the same category after. But the children would only answer whether they saw the same image or if they were different. And unfortunately, what we found with the results for this task is that we see a decrease in the error rate for both groups, control and intervention. However, this is to be expected because this is to be expected if we didn't see any effect of the intervention, because the letter mirroring is a phenomenon that during literacy acquisition will naturally get solved by the process, all of the things that literacy and learning how to write and read comprehend. So what we see here is a decrease that's pretty similar between the two groups. We see here that by the widening of the base of these violin plots, a little, it starts to get lower here in the medians as well. And it was only statistically significant for the comparison between the 2 points of the control group. This could be because of the size of our sample, which is not very big. But still what we see is that the intervention, we didn't adapt the activities. We didn't adapt the activities successfully yet. And then when we correlated the variation of the error rates, we did that only for the intervention group, which is the classrooms, the schools that also had the napping period. So we could only correlate for these students. We found a small negative correlations between the days, amount of days napped and the error rate variation. However, with the p-value of 0.13, we cannot say this is actually a trend. But again, this is an even smaller sample size than we had before. So future studies are needed in order to confirm whether this is actually a trend or not. So thinking about why we didn't see any effect, one of the reasons we believe that led us to that is because in the original protocol, which was done individually, the researcher would guide the hand of the children as a first step in order for them to learn how to write on air or on paper those letters. And then that was done with the children with the eyes closed, and then the student would repeat these gestures by themselves. In our case, we had a situation that we needed to show, the children needed to know what they were doing in the activity, right? This is not a teacher with a single student, this is a teacher with around 20 students. And so for the first week, there was a step in which the teacher showed how, what the activity was going to be like. And that was done with the eyes open, and then the students would do the activities with the eyes closed as we made out to be. We knew that going in. However, all of the possibilities to have the children learn the activities with their eyes closed required materials, and we didn't have funding for that yet. So now we do. So we are very excited to try and do another adaptation of these activities now with materials that will allow us to get to have the children with their eyes closed from beginning to end of the activities, and hopefully find more exciting results from our research. So that's what I had to show today. I would like to thank everybody of Seed Lab. Of course, my friends who, not for only being my friends, but they actually worked with me in this research. And of course, all of the researchers and students involved and that made this project happen. Thank you very much.
Questions? We have time to one question.
Hi, Flora. Thank you. So I have actually a kind of a quick question. So you know the relative amount of time that the child stayed sleeping, and have you ever considered a control in which the child, like you have a group of children that rest but do not sleep, if your intention is to test napping?
So this suggestion has been made before. We didn't control for the children that only rested. We just didn't register that they slept. But this is actually a very good idea, and we will incorporate that for our next version of the protocol.
Well, so thank you. So our next speaker is online, and Estevão, and you are about to connect him. So Estevão, are you there? Estevão? Hi. Hey. Just a second, we are like connecting your audio here. And can you talk? Estevão, can you talk a little bit? Okay.
Yeah.
Can you talk? Acho que tá saindo aqui.
Hello. Hi, hello.
Just a second aqui.
Okay.
Estevão, can you talk?
Hello. Hi, hello.
Just a second aqui. O que vocês acham? Estevão, pode falar novamente, por favor?
Oi, consegue me escutar?
É que a gente tá te ouvindo, só que bem baixo. O seu volume aí tá... Você tá usando o microfone do computador mesmo?
Agora tá melhor?
Ah, melhorou. Fala aí de novo, por favor.
Oi, consegue escutar bem?
Tá melhor.
Ok, sim.
Fala aí de novo, por favor. Agora a gente perdeu de vez. Estevão, fala novamente, por favor. Acho que agora ele não está nem ouvindo a gente. Você está ouvindo?
Acenda aí, por favor.
Acho que não. Não dá só para aumentar o som das caixas? Não. Fala. Oi, Estevão. Testa novamente, por favor.
Alô, oi. Consegue me escutar agora?
Você escuta a gente, sim ou não, com a cabeça, por favor? Sim, ok. Obrigada. Seu microfone está ligado? Estevão, pode falar um pouquinho para a gente testar aqui, por favor, o som.
Oi, consegue escutar?
Vamos só testar mais uma vez aqui, Estevão. Se não der certo, talvez a gente tenha que mudar um pouco o plano e você vai fazer a apresentação junto com o Sérgio. Agora eu perdi o som. Vamos só fazer mais uma tentativa aqui, tá? Senão, eu acho que a gente... Oi, Estevão, tá ouvindo?
Sim, consigo.
Agora vou... Aê, deu certo. Pronto. Pode falar, tá ouvindo, né, Estevão? Me deixa...
Consegue me escutar? Alô?
Sim. Você escuta a gente? Mas se ele não escuta, ele não sabe que vai começar. Estevão, alguém...
So, hello everybody. I hope you can listen to me right now. And first of all, I would like to apologize for not being able to attend in person, but I'm sure that in the next days we can talk or chat there in the institute. So I'm going to present you today a project that we are developing in Sergio's lab that is called the design of a titanium head fixation prosthesis for non-human primates without the use of MRI or CT imaging. So at first, what's the need for the development of such prostheses? In our lab, we perform chronic in vivo recordings of neuronal activity. We basically do that in primates and also in cats, but when we record in primates, we want him to
Estevão?
look at an...
Can you hear me?
Alô? Sim?
Sua apresentação não está carregada. Não estamos vendo. Agora sim. Não está no modo apresentação ainda. Não foi. Agora sim. Pronto. Obrigada.
Ok. Vocês conseguem me escutar?
Sim.
Well, okay, I will start it all over again. So, I will present a project that we're developing in Sergio's lab that is called the designing of a titanium head fixation prosthesis for non-human primates without the use of MRI or CT imaging. And first of all, what's the need for such an implant? In our lab, we perform a chronic in vivo recording of neuronal data that's mainly obtained from primates. So what we need to do? We need the monkey to look directly into an object or a series of visual stimuli that are presented to him on a monitor screen. And at the same time, we do the recording of the neuroactivity, mainly in our case, from visual areas such as V1 or the lateral-geniculate nuclei. To perform such experiment, we need the monkey to have his head fixated so that he won't move along the recordings, and also we have to have a very stable electrode positioned in the correct place so that you have a stable recording and also that you don't damage the tissue beneath it. The classical method of doing such recordings is by positioning a head pulse that is fixated to the skull and also a cylinder through which we position and fix the electrodes. I'm sorry, but I think that I'm not sure if you.
Estevão ficou com...
Tem uma tag aí na sua apresentação.
Bem, eu não sei como corrigir isso, na verdade.
Isso, acho que é melhor deixar assim.
Oi, som, teste, oi. Oi, som. Estevão, você está aí?
Oi, tô sim. Consegue me escutar?
Sim, perfeitamente.
Beleza.
O pessoal está terminando o coffee break.
Tá. Eu recebi uma mensagem que é de ser 16h45 ou é agora? Fico mais atento para agora.
Eu acho que eles iam antecipar o coffee break.
Tá bom. Tá, eu vou estar aqui no computador.
Tá, a gente vai avisando.
Beleza.
Estevão. Oi.
Oi.
Estevão?
Sim.
Ah, tá. Não, é que eu estou testando aqui o áudio, que antes estava com uma microfonia, o áudio estava duplicado, eu estou fazendo o teste aqui, mas aparentemente está resolvido.
Tá bem. Vocês conseguem escutar a minha fala bem também?
Perfeitamente.
Legal, tá bom.
Oi, Estevão. Pode voltar à apresentação? A gente vai dar início.
Ok. Posso mudar aqui?
Ok, está aparecendo.
Posso ir, então?
Eu acho que a Fernanda vai lhe apresentar, só um momento.
Tá bem.
So let's finish the previous session. Our last speaker is Estevão Carlos-Lima. Estevão holds a bachelor degree in biology from UFESP. During his PhD at USP, he investigated synaptic plasticity in neural tissues slices using whole cell patch clamp, with emphasis on neuromodulator effects in the visual cortex. He is now a postdoctoral researcher studying attentional modulation of visual processing using human psychophysics and in vivo electrophysiology in non-human primates. So, thank you, Estevão. And do you have like 15 minutes? If you want, I can tell you when it's missing five minutes.
Yes, please.
Okay. So let's go.
Okay, so thank you, everybody. I hope this second time it works fine. So thank you, everybody, for the opportunity. I will present you a project that we're developing in Sergio's lab that is called the Designing of Titanium Head-Fixation Prostheses for Non-Human Primates without the use of MRI or CT imaging. So first of all, what's the need for such prostheses or implants? When we're trying to record the electrophysiological activity in a chronic method in primates while they are awake, that is, in vivo, we have to have the animal looking or staring at a monitor where we present a different kinds of protocols or tasks. And at the same time, we are recording the electrophysiological activity of some area, in our case, visual areas such as V1 or the geniculate nucleus from the thalamus. The first thing that we have to do is fixate the animal's head so it is staring directly into the monitor so that we can record the eye movements so that we know where he's looking at each step of the recording session. At the same time, we record some electrophysiological activity, and this electrode that penetrates the brain must be very well attached to the skull so that we have a very clear electrophysiological recording, and so that the electrode won't move around the brain and cause any kind of damages. The classical method of doing that is creating some prostheses that, as you can see here, usually there are two separated prostheses, one for the head post, that is for head fixation, and the other is a cylinder where you fixate and tighten the electrodes. To do that, you have to have a very tight connection between the prostheses and the skull. So usually, we create a 3D reconstruction of the skull by using CT, this is tomography, or MRI imaging. But the problem is that usually this kind of imagings, they are very costly, they have limited access and require some level of animal transport. So you have to move your animal from your laboratory to an imaging center and move him back away. So there's always some level of risk while doing that. What we're proposing here is developing a new method of both having the skull measurements and developing a new kind of imaging of implants. I'm sorry, I think that I'm having some kind of audio feedback in here.
A gente tá te ouvindo bem.
So I'm sorry, let me start it over again. So I will present for you in two sections. The first one is the school measurements without the use of any MRI, and a second section will be the new implant prostheses that we develop in our lab. What's the classic method? So you get all these slices, these images from a CT, from a tomography, and you group them together, you mesh it into a single 3D reconstruction of the skull. Although these images are very, have a very fine details, actually the only thing that matters in here when you're trying to do a reconstruction is, let's say that you could simply have two axes designed, and there's a central point between these axes, and you simply have to have the measurement from the 0 point, that is a centroid point, up to the surface of the skull. In this case, we know that is, let's say, 30 millimeters from here or 40, and you just have to have these points so that you can do this reconstruction. So what we thought about was, what if I could do a grid-based reconstruction using a certain number of measurements in each slice, and from that you would design a grid, or that is a matrix, and from that matrix you could do the reconstruction. That is a method without the use of any CT imaging. So let me try to explain this in another way so you can understand our device. So if you do a coronal slice from this skull, you could see those measures. So we would have an alignment point and a centroid, and here is that measure of 35 millimeters. So this is a measure from the inside out, but actually you could have or obtain the same measurement from the external or from an outer method. That is, you just have to have a metric system that starts in the central point, and using a guide tube, that is a measurement from the external point, you could approach it to the outer surface and align it with your metric system. So you would get the same 35 millimeters that you could measure from inside out or from an MCT imaging. So what we need in here is two things. We have to have a system that aligns the skull. In this case, we decided that it should be aligned with the internal auditory meatus, and this is our central point, and we would have to develop some way of fixating these guiding tubes so that they are positioned systematically in the same positions and they are aligned to this rule, that is this metric system. So we designed this external measurement device. As you can see it, it's aligned in here. Let me show you another image of it. So we have our skull model in here. It is, our device is aligned with it, and here is the 3D printed device positioned, inserted into a stereotaxy apparatus. So we can align both the skull, that is the subject, with the device. So we have our central point, and as you can see, through our device, the guiding tubes go through, and we can measure it using this metric system, as you can see here in the image. And our guiding tubes actually are 80 millimeters needles that we can approach from the external part up into the outer surface of the skull. Another important aspect of our device is that it rotates around a pivot point. So you could have different slices, such as just as the coronal slices from a CT imaging. So if we look at this table, you can see the measurements from one of our capuchin monkeys. So we have a grid that is composed of 9 needles or 9 guiding tubes that were positioned in 9 different angles. So we have our matrix that we transformed it into a 3D-based matrix, that is a grid-based reconstruction, that is this black tubes that you can see here in this middle imaging. what we needed now was to have a base template of our school. And we got it from the National Museum of Natural History, a basic template of a capuchin monkey. And as you can see it, obviously the school and our guiding tubes aren't aligned. That is, you have some parts of the school that are not aligned with the measurements from our capuchin monkey. So the simple thing that we had to do was to scope this template into the point that we could inflate some parts of the school and deflate others, so that all the measurements that we got from our capuchin monkey will be aligned with our template. As you can see in here, we have 4 different schools, and they're all from 4 different capuchins from our lab. So it is a very trustful method of recreating or reconstructing the school of our capuchin monkeys in a very low-cost method and without the need of any sophisticated imaging procedure. And also, it's very important that it is a very fast method. In around 30 or 25 minutes, we could collect all these measurements. And what we do, and why do we need to make this? So the main reason for that is for constructing our titanium prostheses. That is, we have our skulls that we created in the software. Now we 3D printed it into a PLA version, and then we forged a aluminum version of it. This aluminum version was used so that we could sculpt or forge the titanium prostheses that we are interested in. This is the design of one of those titanium prostheses that we created. And as you can see, we have here the head post, that is the head fixating point, and the 2 cylinders in here, creating a crowd-like implant. So we have now a single piece structure that is all designed and forged in titanium grade 5, and we forged this in our lab. So also we don't need any CNC method for construction our implant. And also another important aspect of our method is that we don't use any bone wax or acrylic resin. So you can have a higher osseointegration and reduced risks of rejection or infection. Here we have the image of one of our capuchins. This was the first one that we reconstructed the brain and created our implant. And here she is 30 days after the surgery, and so she had a very good recovery. And here she is 5 months post-surgery, and you can see in here the exposed head pin. So it was a great success, all the development of the method and the surgery with this animal. So the key takeaways that we have from here is that we designed a new method of skull measurements that don't depend on CT imaging. It is fast and low cost, and most importantly, it has a low risk for the animal. And also, we developed a new single-piece titanium prosthesis with a higher mechanical strength, higher osseointegration, and with reduced risk of rejection. So I would like to thank everybody for this opportunity. Thank all Sergio's lab members for helping develop this method. And I have to thank also the support from Ciência Pioneira for the support, and thank everybody from the House Symposium. Thank you.
Thank you, Estevão. Questions?
Sorry, Fernando, that's your job. So, Estevão, thank you so much for the talk. There's a bit of an echo here. So, what is the precision of the angle and the distance measurements that you get? Or what is the error expected?
I'm sorry, I'm not sure if I could get the question entirely. What's the precision of the angle? Is that it? So if I could go back to the slides, you can see that we have these pins over here. You can see it better. So each step that we fixate in here is a 12 degrees angle. So we have 9 steps in here that we can rotate, and it's always positioned in the same place. This is one of the schools that we have to test with, and we've done it like 5 or 4 times with the same school before we started working with our animals, and we always got exactly the same measurement. We had an error of 1 or 2% in each measurement, so it is actually very precise. We've always worked, we developed the method for working with our capuchin monkeys, but now we're trying to develop it now, a new version to work with cats, and also we hope that it works fine with rhesus monkeys.
Do you have any evidence of osseointegration?
I'm sorry, I couldn't understand the question.
So thank you. Tá falando? Thank you, Estevão. And yeah, so thank you for the presentation. Bye.
Thank you, bye-bye.
Okay. I think he's still online. Thank you, Estevão. Thank you very much for this nice talk. And now we are moving to the last speaker of our symposium, and it's a pleasure to welcome Fred Wolf from the University of Göttingen and Max Planck Institute for Dynamics and Self-Organization. Fred is a long-term collaborator, and I know him since a couple of years. And he's a theoretical physicist who studied in Frankfurt, and he focusing on complex system physics. And neuroscience was part of a big physics research project in Frankfurt at that time. And he was the first physics students actually who did kind of a major or an emphasis in neuroscience, which was particularly mounted by Wolf Singer at that